Harmit is going to be talking on viruses, and both plants and animals suffer from viruses, so this can be considered a lecture in either botany or zoology. Our lecturer, Harmit, did not grow up on a farm in Felicity, Ohio, nor did he suffer from extreme childhood allergies. Rather, he got his bachelor's degree from the Institute of Technology in Bombay, India, in chemical engineering, and then he went to the University of Rochester in upstate New York to do a PhD in biology, partly because the University of Rochester would accept somebody with a chemical engineering degree into their biology program. From there, he moved on as a postdoc to the Fred Hutchinson Institute in Seattle, and they recognized when they were on to a good thing because he's been there ever since, despite occasional attempts by Harvard to woo him to Boston as a much more clement climb than Seattle. In Seattle, he is in the Fred Hutchinson Cancer Research Institute. He's also an adjunct professor in the Department of Genome Sciences at the University of Washington, and is an investigator at the Howard Hughes Medical Institute. So, Harmit Malik.
Thank you very much, David, and thank you everyone for being here. It's a real honor to be giving the Prather lectures this year. It's a little bit intimidating to hear that this is the most prestigious lecture at Harvard. So I'll try to do my best to not make this in biology. Thanks for clarifying. I have to say that it's a little bit daunting to give a lecture that's designed for, like, if I'm representing botany, this is going to go very badly. Because I think the last time I successfully grew a plant, it was a cactus. So that's sort of the limits of my botanical expertise. I'm also going to make the suggestion, if it's okay with the audience, that I would actually encourage you to stop me in the middle of my lecture to ask any questions that come up. This is meant to be somewhat of a public lecture, but it's also, I would love for it to be interactive. And I'm sure that if you have some questions, it reflects the fact that I probably didn't explain something well, and other people probably have the same question. So there's no pressure, but if you have a question, please shout it out. And the material is designed to incorporate questions in the period, so don't feel weird about that. So I kind of want to start my lecture with a picture that many of you recognize, at least from your tourist expeditions or at least from geology terms. This is, of course, a picture of the Grand Canyon, and this is one of the prettiest pictures that I have found, so I like to show it over and over again. But I sort of use this picture to emphasize what we know about geological time is really basically from features that are crafted into the Earth. And yet there was a lot of sort of controversy about how old is the Grand Canyon. So when I was going to graduate school, estimates were sort of in the 60 to 70 million year range, because people thought that that was basically the age of the Colorado River. But as more and more evidence has accumulated, it's actually clear that it's far younger than that. The Grand Canyon is probably only about 6 million years old, even though the Colorado River has been probably carving canyons for much longer than that. So this is kind of incredible, because if you think about the Grand Canyon and the entirety and the depth of the Grand Canyon, 6 million years is a lot of time, but it's also like almost no time when you think about it in the context of the history of Earth, and specifically the history of life on Earth. So one of the sort of starting points for this lecture tonight is for us to just briefly contemplate what that number means, which is about 3.5 billion years since life has existed on our planet. And rather than think about this as sort of a collection of years, I would like you to sort of really remember that this is not just a collection of years like grains of the sand on the beach, as this quote points out, but it's really like a series of years, one following the other. And it is actually that series that has allowed the diversity of life as it exists on the planet to really even be possible. It's basically this collection of the sort of history of life on Earth. And sort of my favorite quote is that all those years have really passed moment by moment, one by one. It's sort of powerful to think about it that way. They encompass an actual already lived reality, encompassing all of the lives of all of the organisms that have come and gone in that time. It's that expanse of time that defines the realm of biological possibility in which life and its extraordinary diversity has evolved. It is time that has allowed the making of us. And it's really sort of important for us to consider, but there's lots of sort of debates that go back and forth, probably not in this audience, about evolution and how is it possible that we have all of this complexity. But there's very little that the human brain can sort of comprehend with this very, very large number in mind, and just the possibility that that number affords to life on the planet. So today I'm actually going to tell you about one of those possibilities that comes about from a sort of different type of geological record, which is from ancient viruses. I'm going to sort of begin my lecture with this plate of the mummified head of Ramses V of Egypt, who is obviously one of the greatest sort of archaeological finds in history. But one of the really cool things when this mummy was discovered was actually the finding from medical archaeologists that almost certainly King Ramses was infected with the unmistakable signs of smallpox and likely died of a smallpox infection. And we know that this is possible because this is at a time when smallpox was raging through many parts of Egypt and actually large parts of the world. So in our sort of history, in recorded human history, there have been records of this sort of epidemic scale, which has really affected the human species as a whole. And here's another much shallower record of a virus that has affected the human species as a whole. This is, of course, a slightly out-of-date prevalence of HIV among adults aged 15 to 49 in 2016. And sort of the really sort of important and sobering message to sort of think about is that all of this history began with one cross-species transmission event. That is, you can debate like exactly when it happened, but it is by no means older than 150 years ago, right? So 150 years ago, there was a transmission event, and there have been subsequent transmission events, but there was one prototypic transmission event that led to the pandemic as we know it today. And there are millions and millions of people who have died. And if you sort of just think about the staggering influence of one of these zoonotic transmissions into the human species, this gives you the sort of sense of the power of a pathogenic virus that sort of invades a sort of naive species and has spread. We are lucky that we live in a world today where there are mechanisms of control with antiretroviral therapy. But when this statistic was actually diagnosed, of course, these therapies had not reached parts of sub-Saharan Africa. And particularly in some of these parts, life expectancy on average in males was less than 27 years. And it was completely driven by one sort of pathogenic virus. So it's a really sort of important and sobering reminder of how one sort of pathogenic virus can actually really have such a profound impact. And of course, smallpox had this impact before. So our history is recorded with these kinds of anecdotal episodes of viruses that have the capacity of sort of really, in a pretty dominant fashion, shaping human evolution. We have young viruses like HIV and smallpox. We have older viruses which we thought were under very good control, but because of sociological reasons are making a reappearance, which really sort of disturbs virologists like me. There are other viruses that we really deeply worried about that actually never really took off from that initial sort of signature of looking like they could become a pandemic. For instance, the SARS coronavirus. But this is sort of not an ended reality. This is part of a continuum of viruses that continually challenge us. And with air traffic being what it is, this is actually not now limited to sort of spread as being the primary determinant of what would happen with the virus. I'm actually not going to talk about these historical viruses per se. Instead, I'm actually going to talk about even older viruses for which the historical record is not from recorded human history, but it's actually from the remnants of these viruses that sit in perhaps the most pristine record of these viruses, which is our own genome. So these are fossilized viruses that exist in the human genome and really highlight a history of these viral infections as they've occurred over millions and millions of years ago. So just to get everybody on the same page, I recognize there are both biologists and non-biologists in the audience. I just want to go through one class of viruses that I'm going to focus the bulk of my talk today, which are retroviruses, which include viruses such as HIV. So this is just a cartoon of what the viral life cycle looks like when a virus is actually infecting a cell. So the virus comes in, it engages with a host receptor. Based on that docking, it'll basically come in and reveal basically a viral capsid, which further undergoes life cycle. This capsid is disassembled, and there's a very critical step where the RNA genome of this virus is actually reverse transcribed into DNA. This is actually against what was then considered the central dogma of molecular biology, and led to the Nobel Prize being awarded for the discovery of this process. And then that double-stranded DNA is actually integrated into the host genome. And it is that integrated virus, which is called a provirus, that is actually used to produce all of the material needed to produce the daughter viruses. So it's not that you're going from a viral virus to virus. You're actually going from virus to virus via an obligate step that requires integration into the host genome. So what's interesting about that is that even after this provirus has stopped being able to produce daughter viruses, it still sits in the genome as a reminder of that event that happened when that retrovirus first invaded. Which means that we can actually think about viruses as having now two ways of transmitting to the next generation. So let's consider these cuddly koalas here. They're basically going through what is a pretty considerable invasion by an exogenous virus. This is an ongoing epidemic. And the typical way we think about this is that these are transmitted from one individual to the other individual. Typically, this is by sexual transmission, but it could also be by some sort of biting mechanism. But at some rare frequency, these viruses can endogenize, which means they will integrate into the cells that produce the egg and the sperm for these koalas, and therefore be transmitted to the next generation. So now this mother koala has two means of actually transmitting the virus to the next generation. One means is obviously via the infectious route, but the other means is actually by transmitting these imprinted viruses that are in her genome and now passed on to her progeny. So these are what we refer to as endogenous viruses. And these viruses are often completely capable of reinfecting new individuals by becoming exogenized. So essentially, she has, as part of her legacy, transmitted viruses in two forms to her progeny. And the longer they sit in the genome, the more likely it is that they're going to get more and more prevalent in the population, and therefore basically become fixed in the population as an indication of what was this ancient viral attack at one point in history. I chose koalas in particular because they're sort of the closest example we have to what must have been the HIV infection or invasion of the human genome. Because about the same time as HIV was entering the human population, we estimate that this koala retrovirus actually entered the koala populations in the northeast of Australia, and over the last 100 years has basically marched down the coast of Australia. When this paper was reported in 2006, there was really only one population of koalas that was protected from the koala retrovirus. And this is a pretty nasty virus. This is not something that you would basically want to get if you're a koala lover. Unfortunately, this is no longer the case that this is quarantined, and that's thanks to mostly human tourists, where basically now this virus has essentially made all of its way here. Now, what's really interesting is that these koalas are separated temporally, right? Because one of these populations got infected much before the other. So they're also separated in terms of the imprints made by the koala retrovirus in their genomes. So for instance, these koalas have typically on the order of 100 independent imprints of the koala retrovirus in their genome, whereas these koalas have barely any because this is a very young invasion in this population. So think about this, right? So a typical retrovirus is about 7 kilobases in length, or 7,000 base pairs of DNA units. There are about 100 units of retroviruses, which means 700,000 bits of information of DNA, which are all derived from a retrovirus, that distinguish this koala population from this koala population, even though they were basically the same species like 150 years ago. So this is, again, a remarkable example of how the sort of invasion of a retrovirus can quickly distinguish the sort of genetic heritage of what these species kind of look like. We have basically the same imprints, but just magnified many-fold. So in our genome, we have had waves upon waves of retrovirus infections. The sort of thickness of the arrows is just to give you the best order estimate about the intensity of these retroviruses that have invaded the human genome. The last wave of active endogenous retroviruses that became endogenized in the human genome, we estimate, was about 600,000 years ago. So we've not had any new action. And even though it's a slightly controversial thing to say, because there's some sort of data that suggests otherwise, the consensus is that there are no human endogenous retroviruses that are still capable of basically jumping or becoming infectious. That is certainly not true in most of our primate relatives. In most of our primate relatives, they harbor in their genome retroviruses that are fully capable of still being capable of producing infectious retroviruses. So what is the bulk heritage of this window where we can basically see these waves upon waves of retrovirus infections in the genome? So would anyone want to hazard a guess? Like, how many such imprints would we be able to tabulate in the human genome if we were to just go through as an actuarial person? 5,000. That's a good kind of starting estimate. 5,000 is a pretty big number, though, right? Like, but we're talking geological timescales. Does anybody else have a guess? We're going to play the Bob Barker game, or? So actually, it turns out that it's about 100,000 indicration events from at least 31 different classes of retroviruses, which means that even if you were to estimate as just a minimum number, the minimum number of events where a retrovirus infected an individual, not just the individual, the germline or the sperm and egg producing cells of that individual got passed on to the individual and passed on to all members of that species eventually. So each of these are extremely low probability events. I hope you can appreciate that. 31 times this has already happened at a minimum. And it's quite likely that many of these retroviruses invaded multiple times, but we don't have the temporal record to separate that. So that seems like a lot of DNA, right? Like, so that's 100,000 independent events of retroviruses inserted into our genome. And at a minimum, that's about 8% of the human genome as it exists today. That actually didn't exist until 60 million years ago. So this expansion in bulk of the human genome has been entirely driven by these waves upon waves of retroviruses sitting in, making a home as they go about their life cycle, and then being passed on as an imprint of that invasion from one generation to the next. That's a lot of DNA, because it actually turns out that maybe less than 3% of your genome is actively producing the RNAs and proteins that we can ascribe to actually human function. In fact, 8% is these fossil virus records. I'm not even going to talk about the nearly third of your genome that is made up of a completely different class of selfish elements that doesn't even have an infectious phase, but actually goes through its merry little life cycle of selfishness within our genome. It's also important to point out that 54% of the genome remains mostly undescribed as to what it is, but most of us feel quite sure that that actually represents viruses and elements that have decayed so much that we can't actually fully describe where they necessarily came from. So this is even an underestimate of the total bulk of viruses that sits in your genome. Now, if this was your IT department, you would totally fired them, right? Because this is a hard drive that is full of spam and basically viruses, right? Like, that's basically the pristine state of the human genome that exists today. So it's really important to sort of think about what the bulk of this virus is, but maybe I want to give you a little bit of insight into how do we do these kinds of analysis and what makes it kind of possible. So we've already alluded to this. So here, let's consider a a typical retrovirus in its sort of simplest form. This retrovirus has 3 types of genes. There is a GAG gene, which makes up the capsid of the retrovirus. If you were to imagine from a Star Trek analogy, this is what makes up the bulkhead of the spaceship, right? The polymerase gene is all of the enzymatic functions encoded by the virus, or if you will, the software that makes all of the spaceship go. And the envelope gene is the shield of the retrovirus that actually both protects it from the immune system, but that's actually what is used to dock with receptors and gives this retrovirus the ability to infect one cell from one cell to the other. So this is the sort of all of the mechanics of what makes the retrovirus work. And as I've already told you, an obligate step in that life cycle is that provirus will basically sit in the genome and spew daughter viruses until it's incapable of doing so. But even after it has lost the capacity of producing daughter viruses, it's not going to be evicted from the genome. There's no mechanism to do that. So the longer it sits in the genome, the more it's going to be subject to just random mutations, just like any other part of the genome. And because this retrovirus is basically not a useful thing for the genome to keep around, it's going to accumulate more and more mutations. And the longer it sits in the genome, the more mutations it accumulates. And so because of a very nice quirk, these viruses happen to be surrounded by 2 terminal repeats that upon insertion are 100% identical. So based on the divergence of these repeats, we can very precisely date the age of how long that particular virus has been sitting on the genome based on the types and numbers of mutations it has accumulated. Now, of course, we have an even better record because we can actually look in the genomes not just of humans, but all of our primate relatives and date when the viruses first came based on their precise location in the genome. So in a way, if you were to construct a phylogenetic history of our primates and leading up to humans, based only on this record of these dead viruses, you would precisely recalibrate all of primate evolution just based on this beautiful fossil record of viruses and when they came into our primate history. This is pretty much as boring as it sounds. I don't want to make it sound more glamorous. Like this is like an insurance agent scanning through decades of like tax documents, hoping to find the one mistake that Al Capone probably made. And most of the time you don't find this mistake, so you just sort of garner just bad karma, right? Because you're just going through the stuff. I spent many, many years doing this as a graduate student, so I definitely don't want to make it more glamorous than it sounds. But once in a while, you discover something that kind of makes you just scream out in disbelief. Because, for instance, we have some cases where in the midst of this dead provirus, we have a gene that is basically completely resistant to mutation, almost as if at one point in evolution, the human genome decided, or the primate genome decided, well, that's a gene I'd like to protect from mutation, so I'm not going to get random mutation, get rid of this gene, because this is something that might be actually useful. Of course, I'm anthropomorphizing what natural selection looked like, but this is basically like saying a retroviral insertion landed in the genome, 99.9% of the time that'll decay completely, but at a very small fraction, some fraction of that retrovirus is prevented from mutationally decaying. I'm actually going to spend a lot of time thinking about why is it that you would want to domesticate the envelope gene, because as I've told you, the envelope gene is the very gene that gives retroviruses the ability to be infectious. So this was even more puzzling to consider that the envelope gene is actually what has actually been preserved. So the prototypic case, which was identified many years ago in Jim Mallet's lab, different Mallet for those of you who are thinking, was that the big clue actually came from the finding that this gene called syncytin appears to be most abundantly expressed in the placenta, right? So it's a 35 million year old dead virus, completely incapable of jumping. One of its genes is preserved. This is the envelope gene. And that gene is most abundantly expressed in the placenta. Now, what's really interesting is it's been sitting in the host genome or primate genomes for nearly 35 million years. Yet you can take the gene as it sits in the human genome and splice it back onto a backbone of a retrovirus in a cell infection experiment, and it's perfectly capable of still acting as a retroviral envelope gene, right? So it's not lost any characteristic features of its ability to cause infections. So we have what appears to be an infectious causing protein sitting and abundantly expressed in the placenta, which is kind of cool, but also a little bit bizarre, until I sort of remind you that the placenta is a very, very special organ. In many ways, perhaps the most special organ as far as mammalian evolution is concerned, because it's extremely diverse. It's an organ that is clearly derived from the fetus, but it is actually the only organ that directly interfaces with mom in terms of nutrition transmission, but also negotiating the intricate immune reactions that take place when you have a foreign object growing inside mom. So experiments in a number of labs have shown that syncytin is basically exclusively expressed in these syncytial trophoblasts, hence the name syncytin. And these syncytial trophoblasts have the very special property that they undergo cell fusion in order to actually mediate this nutrition transfer from mom to fetus. They also have this very interesting property that they can potentially protect the fetus from the unfettered attack of mom onto the fetus, which is a different organism growing inside her. It's interesting because retroviral envelopes have those two features in common. They have the ability to mediate cell-cell fusion. That's, after all, how they become infectious. And they have this immunosuppressive domain, which is what actually allows cells that are infected with this retrovirus to not be attacked by the immune system to recognize them. So it's really kind of important to consider that it is possible that, at least in primates, our ability fundamentally to do biological reproduction by live birth via the placenta might itself have derived from what appears to be a retroviral envelope gene from a retrovirus that has decayed but landed in our ancestors about 35 million years ago, right? Are there any questions about this?
What do other placental mammals do?
Yeah, I'm going to get to that in the next slide. Let's hold on to that talk. Anything else about this? I welcome questions. I'm not deferring. I'm just going to talk about it on the next slide. Yes, Ethan. Yeah, excellent question. Are there women who are unable to? We don't know of any women that are homozygous defective in syncytin, but we can definitely engineer these in other mammals, and I'm going to get to that. So let's get to the sort of question. I've told you this kind of amazing and probably slightly unbelievable story where, like, a fundamental feature of our biological reproduction is like one of these spoils of war from retroviruses that happen to land in our genome. But this is all primates, right? But that's a pretty big span, right? 35 million years of simian primate evolution. But of course, primates are not the only mammalian lineage that has placentation as the primary form of biological reproduction. What about rodents? Or what about rabbits and hares and sheep and goats and cats and dogs? It turns out, rather remarkably, each of these lineages has their own syncytin. Each of that syncytin derives from a completely different retroviral envelope gene. It's the most remarkable example of lightning striking 7 times during mammalian evolution, each providing each of these lineages an active envelope, which is basically being used to mediate these infection experiments. In rodents, where is the one place which has been investigated, there are actually 2 active syncytins. Each of them works in a different layer of the placenta. One that's immediately proximal to mom and one that's immediately proximal to the fetus. If you knock out even one of these genes, you lose the ability to sustain pregnancies to term. So this is basically an essential gene in every sense, if you think about it in the context of mammalian reproduction. Yes?
Do you have any sense of what served the same purpose before 35 million?
Yes, excellent question. So this is a little bizarre, right? Because they've been doing placental birth for longer than the question is, what were they using before the last 35 million years? So our sense is that this is very much like trading up. So what we are looking at today is the present day versions of syncytins that are being used because they're actually protected. But we have evidence from our own genome that we actually had an even older syncytin that we've now traded up by getting a younger syncytin in the genome. So essentially, because retroviruses keep landing in our genome, we are afforded more and more opportunities to take what is an active under-selection envelope. And so given the choice between a syncytin gene you already have and a new potential syncytin gene you come in many cases you just trade it up, very much like you trade a new car and you trade a new car for an older car. So we think that there was a prototypic syncytin, And that prototypic syncytin has been replaced in these waves. Because the prototypic syncytin just now looks like a decayed retrovirus, we don't actually know what the prototypic syncytin is because there's no way to distinguish it from the 100,000 other integration events in the genome. But that's the most parsimonious explanation for all of this. Now, importantly, I don't have marsupials on this, which are also placental, because that's actually not nailed down yet. There is a very, very good candidate. This is work being mostly done in Terry Heidman's lab, and he's actively on the case. But we don't have that, so I'm not going to say that we know what marsupial syncytin looks like. But there is another mammal that is not on this lineage, which the astute among you should know, which is platypus, right? You guys have seen some version of this before. Well, platypi are really, really cool because they're the only egg-laying lineage, right, of mammals. And I'd like to remind everybody that long time ago when mammals broke off, our ancestors were birds, which are also egg-laying individuals. And in fact, platypi represent this history of egg-laying in the mammalian lineage. But then we switched in this dramatic switch from egg-laying to live birth in mammals. And that switch actually coincided, we believe, with this gain of syncytins across different lineages and multiple times. Yes?
That does mean that reptiles that do oviparous can also do, like snakes?
Yes, so hold on to that question. Not all of them have placenta, which is like a very specific thing, but I'm going to talk about one that does in the next slide, if you just hold to that question. Now, here's the thing. This is sort of a very good hypothesis that fits the bill, right? We went from this transition from egg laying. What is the other piece of evidence that we have? Now, egg laying requires a cascade of genes called vitellogenesis genes, which are really responsible for producing the eggshell, et cetera, totally conserved in birds, also conserved in platypi. Amazingly, you can find remnants of these egg-laying genes in our genome, but they've been dead because they've not been used for 180 million years. But because these are long genes and we can identify these remnants, it's very, very clear that we had these genes, and these genes are active in platypi, but not active in the human genome. And that's a very good historical record of the history of our ancestry as basically egg-laying organisms. Now, we won't get into the mechanics of why this was a good thing or a bad thing. There's lots of debate about that. But just consider the alternate reality in which instead of going into sort of delivery birth rooms, we'd basically go into these growth chambers like chickens and lay eggs, and that's kind of how birth would happen. This is a different thing to contemplate. Somebody asked me about reptiles. So actually, placentation is actually not unique to mammals, which is sort of shocking in a way. There are many fishes that undergo placentation. We actually don't know the basis for this, but there's people actively working on this. But this one is like really cool because this is a lizard called the Mabuya lizard, which is the only reptile lineage that we know of that actively does live birth via placenta. And so that's pretty bizarre, but basically it's a great evolutionary experiment, right? Like, well, how did this placentation occur? Well, it turns out this was also a syncytin that basically came in and is now expressed in the lizard placenta, where people have shown that that is actually directly responsible for the placental sort of live birth mechanism in lizards. So this is pretty much as like close and open and shut case as evolution gets. Like you basically need this mechanism. I want to make it very clear this is necessary but not sufficient for placentation because there's a whole gene regulatory network cascade that needs to happen to have this sort of dramatic shift. That gene regulatory network cascade also derives from a lot of these retroviruses, but we're not going to talk about that today since we're focused on protein coding genes. So the real smoking gun evidence would be what would these fish do that are basically capable of undergoing placentation, and that's actually work being done in a number of labs. There was a question? Yes?
Yeah, where would the viviparous sharks fit into that? Because they've been around longer than...
Yes, well, the sharks would definitely be a good one, except those don't undergo placentation. So it's a different form of basically providing nutrition through a live birth. And so that's a very specific example of like how you do live birth. And so there are a number of viviparous individuals that basically do different forms of reproduction. Also cool, but doesn't tell us anything about the history of placenta. Yeah, excellent question. So I just don't want you to leave with the impression that actually these genes sit in our genome because they're a form of biological reproduction. I sort of want to remind you that the primary purpose of the retroviral envelope gene is to negotiate interactions with host cell receptors. These could be calcium transporters, anything that's sitting in the surface of the cell, and basically mediate the entry of the virus into the cell. That's the primary purpose of the envelope gene. They provide the mechanical energy as well as the docking required for this. Now, as these genomes sit in the genome, we've also encountered domesticated versions of retroviral envelope genes. These are, again, just like syncytin, expressed in the genome. So you could imagine them as being evolutionary turncoats. They started life as a retroviral envelope gene. Now they sit in the genome, and the host genome is now weaponizing them for their own purpose. So one very simple way for these domesticated genes to be useful is for them to continue doing what they've been doing, which is engage with the receptor and undergo endocytosis. Why is this important? Well, it's important because now when the retrovirus comes in, it has nothing to engage from. You've effectively created a receptor loss experiment. Now, that's not necessarily a great thing, right? Because you've now got rid of the receptor, but you've done it in a controlled fashion, and it basically now allows this organism to be protected from this retroviral envelope. Now, the really cool thing is these domesticated genes are basically kissing cousins of these retroviruses. So these are often very young events, very young domesticated genes that have the capacity, of being protected. I apologize that you have to go through the animation again. But I just want to emphasize that this has also happened at least 3 times in evolution. One of the most famous examples is the FE4 gene in mice, but also in chickens and in sheep. In sheep, this is an active, ongoing infection called the Yagti sheep retrovirus. These envelope genes have the capacity not just to engage with the receptor and get rid of it, but they also have the capacity, being expressed, to train the immune system so that it's actually primed for when the infectious retrovirus comes in, it can clear it out. So it's basically another way of adaptively priming the immune system to prepare it for whatever retrovirus actually comes in. We are actually also studying cases of retroviral infection in my lab actively now, but we are actually studying them in individuals which, to our best of our knowledge, do not have placenta. But we are very interested in them because some of these insertion events, and they've been preserved for more than 200 million years of evolution, right? So they're kind of deeply profound. They've never been lost as far as we can tell. And they're not expressed in some exotic tissue. They appear to be expressed everywhere as far as we can tell. So we're very interested in understanding what is the new biology that's actually conferred by these types of individuals. So the stories I've been telling you about have been about the envelope gene. It's kind of really cool because these are the infectious associated genes. I don't want you to give the impression that this is one-off cases. There are other cases which still sit in our genome, which we have no idea what their function is, but they are clearly preserved or have been preserved for millions of years. So there's active areas of investigation still ongoing. We have no instances of the enzymatic functions of the retroviruses that have been preserved. Now, there are enzymatic functions from other cellfish elements that have been preserved, but none from retroviruses, which is really bizarre because these enzymatic functions are things like proteolysis, reverse transcriptase, totally cool new function, but they haven't been domesticated. So it's interesting to consider that the envelope gene would be domesticated so many times, but not the enzymatic functions. But the granddaddy is the capsid genes, right? We've known for a very long time there's lots of capsid genes, some of which date back at least 100 million years, some date back more than 250 million years, that have been sitting in the genome, totally protected from mutation, just like a human gene would be. But we really didn't have a good idea of what the function of these would be. And that all changed rather dramatically in 2 excellent papers that appeared last year, one from Jason Shepard's lab and one from Vivian Budnick's lab at UMass in Worcester, in case somebody's here. And these all came from totally kind of non-directed findings from neurobiology, where people had been studying this ARC gene, because this gene is actually important for establishing synaptic plasticity and neuron-to-neuron communication, and interestingly, has been studied for that function in both Drosophila and in mammals. The first identification of the ARC gene actually came from human patient studies where people came into the clinic because they had clear neurological deficits, and they were basically tracked down to a compound heterozygote, for those who you know, in the ARC gene. So we knew that that gene was really important. The ARC gene actually is basically sending pockets of information from one neuron to the other neuron. And they're doing these in what looked very, very suspiciously like viral capsids. Now, interestingly, the only cargo that we know that the ARC genes are sending across is the ARC transcript itself, right? So it's almost like you're transmitting yourself across neuronal boundaries from one to the other. People strongly suspect that there's other cargo in here, but we don't know what that is. And this transmission is basically very much like a viral infection cycle, except you're not reliant on the envelope anymore. You're using other mechanisms of membrane-to-membrane transmission. The reason I'm spending so much time telling you about these ARC genes is because these ARC genes are all gag genes. And in fact, in two separate domestications from these retrovirus-like elements called gypsy, the gag gene has been domesticated a very long time ago to lead to the ARC gene in mammals, and a separate gypsy element gave rise to a completely independent domestication that has given rise to the ARC gene in Drosophila. Both of these genes are completely essential for neuronal transmission. And there's at least 30 more genes like ARC that remain kind of uncharacterized, right? So if you just sort of consider the inherent complexity about what happens with this transmission, it's worth considering it might be a really good idea to start basically going through each of these domestic gag genes one by one to basically look at their functions. The problem is that some of these probably have redundant functions, which is why we've not been able to ascribe function. What makes these genes special is they don't have a non-redundant role, which is why we were able to identify them in screens. I should point out that I actually spent, as a graduate student, a lot of time studying the Drosophila ARC gene, as it turns out, and I even have a thesis chapter. I had no idea what it did. I said, this is really curious. It's been preserved for so long. So it's really nice to see that sort of ARC ending, no pun intended. Okay, so this is an example of all of this sort of new biology, really important biology in our genome that basically comes from these totally stochastic events like meteors landing on the surface of the moon. These retroviruses landed in our genome, and some part of that retrovirus was deemed so important that we basically protected this from degradation. So it's really worth considering how much of our biology is potentially shaped from these stolen genes. And this is kind of like really the main take-home message that I'd like you to consider, is that we sort of think about this, our biology, as this sort of unitary inheritance from a common ancestor. But we have all kinds of stuff landing from different sources, including from bacteria, including from viruses. And they're not just sort of happenstance events, they're actually leading to new biological transitions in pretty fundamental mechanisms. Retroviruses are only the tip of the iceberg, and it's really, really important for me to emphasize that. Retroviruses are unique among viruses in the sense that they need an obligate step in their life cycle where they make an imprint in the host genome. Other viruses don't do this. They can go virus to virus without ever making an imprint. But nonetheless, because they're going through their life cycle within the host cell, there is a very rare possibility that their RNA can be used and integrated back into the host genome via reverse transcription reaction. So our knowledge of these non-retrovirus viruses is basically like winning the lottery. Some virus went through its life cycle at some point in evolution and was used to basically integrate that particular viral fossil in our genome. But that is still a very rich history when you consider all the genome sequences that we have. So for instance, in our own genome, we have imprints of Borna viruses that go back at least 60 million years, and in some cases still are capable of encoding genes. We have no idea what these do. People have strongly suggested on, I don't want to offend anybody, somewhat questionable evidence about their link to schizophrenia. There is some sort of evidence that that might be the case. And so people have suggested that these might also be protective against those kinds of viral infections. We have a 35 million year imprint from a filovirus. Does anybody know, non-biologists know what a filovirus, a famous filovirus is? Ebola, right? And Marburg. So Ebola is not a young virus at all by any means, right? 35 million years ago, because it sits exactly in the same spot in our ancestors as well as in us. An imprint came in. So not only did we encounter an Ebola-like virus, we basically survived it, right? That's why we are all here. We all carry the imprint. So this is not just a record of the viral interactions with our ancestors. It's also basically a record of the fact that we were able to fight off these pathogenic viruses in order to actually leave children to carry the imprint in the next place. I sort of want to point out that this is actually, as we get more and more data across the mammalian lineage, we are getting more and more deep knowledge about these viruses and how they basically, at least a historical record of these viruses. We used to think that these viruses are tens of thousands of years old. Now we know that many of them are 100 million years old. In particular, for instance, there's this Ebola imprint that is common to all bats. That's kind of incredible because that lineage is 105 million years old at least, which means 105 million years old, an Ebola imprint landed in bats, and there's a gene from Ebola from that imprint that is totally preserved. And people are actively testing whether that imprinted Ebola gene is basically being used as defense along the lines of what I showed you before. And maybe that's why bats basically are not so dramatically affected by Ebola pathogenicity. So there's a lot of projects here in terms of understanding this new biology that is probably shaped from this happenstance event. I don't want to give you the impression that these are all active genes. Most of these are not active genes. They're simply a historical marker of a viral infection that happened at this point in evolution. So I'm just going to end my talk by talking about what my lab is doing to try to leverage these past infections. So one thing we've learned is that each of these infections triggered an episode of adaptation in the genome to try to fight off these pathogenic viruses. And that led to very rapid evolution of our immune genes as we basically went through these cycles where the immune gene tried to evolve to block the new viral protein. The viral protein evolved very rapidly to escape that immunity, leading to very rapid evolution of our immune genes. In fact, if you were to line up almost like in a speed limit kind fashion, all the genes and their rate of evolution, immune genes are basically complete scofflaws in terms of their rate of evolution. They are breaking the speed limit over and over, but they really need to because it is their rapid evolution that is allowing us to keep pace with the changing pathogens that we encounter over the course of evolution. So what we are very interested in is asking, what can we learn from these kinds of arms races? So one thing we've learned is that a lot of this arms race is actually happening at the direct interface at which immune genes interact with viral proteins. So here's an example of an immune protein that we'd like to talk about. So essentially what we're doing is we're bouncing between these states in which the antiviral protein is able to engage with the viral protein, so the host is winning, or the virus is able to evade this binding affinity in case the virus is winning. And we've gone back and forth over the course of evolution with multiple different viruses to play exactly the same game, chiseling away at the interaction interface between these two components. And we identify these interaction interfaces by looking at where the rapid evolution can be concentrated on protein domains. It's not important for you to know what this protein is, but it's a very important antiviral protein called MXA, which was my lecture yesterday. And just based on the very rapid signature of evolution, we've been able to pinpoint or highlight potential interaction interfaces where this protein is engaging with a complete history of ancient and current viruses. And what another graduate student in the lab is doing is basically taking that same history of rapid evolution and trying to now manipulate that history in the lab. Basically take the lessons from evolution that we have learned in terms of human immunity evolution and try to now accelerate that evolution in the lab to try to identify new immune genes or new immune variants of existing genes. So here's just an example of something we did recently where we took the wild-type human MXA protein against an influenza-like virus called tagotovirus. You can see you have a pretty big boost over a catalytically dead version, a 10-fold increase. This is what a typical very good antiviral response would look like. And just by manipulating 5 sites under very rapid evolution, we've been able to elicit in the lab variants of this immune gene that are 10 to 20 times better. So what we're doing is we're taking the rules of evolution, but also taking the advantages of DNA synthesis and our ability to do combinatorial mutagenesis to basically potentially accelerate, but only in a very tight window, evolution of these interfaces to try to elicit even more potent versions of these antiviral proteins. And if you're able to do this successfully, in theory, we could actually apply this to a general study of antiviral genes and elicit variants that could go after pathogenic viruses that our immune system is currently incapable of defending. And with that, I just want to end my talk by thanking my colleague, Michael Emmerman. So Michael Emmerman is a virologist who spent his entire career studying the HIV virus. He was actually he trained in both labs that identified different versions of HIV that have entered the human species. And at least when we met, he was not an evolutionary biologist. And when we met, I had never done a single experiment in virology. But we basically just really hit it off and realized that studying this sort of ancient viral record is a really good way for us to essentially describe what has happened deep in evolution, but also describe what might be going on today. So I owe him a big debt of thanks. And thank you very much.